Hello, welcome to module 5 of 4th week of our course. In this module, we will build a model for a small genetic circuit as we have done in the earlier module. Let us look into the circuit first. In this circuit, we have three genes, X, Y and Z. In the earlier one, we had two, X and Y. Now, in this module, we will have a circuit made up of three genes, X, Y and Z. In the earlier module, where we have built a repressor of repressor, so X was repressing Y and Y was repressing X. In this case, X is repressing Y and Y is repressing Z and Z in turn is repressing X. So, I have X repressing Y, Y repressing Z and Z repressing X. So, it is a mutual repression involving three different genes. Each of them can be considered a transcription factor and each of them control the other one, X, Y and Z in a series. This type of circuit has been created artificially in E. coli and other systems, and their dynamic has been studied both theoretically as well as by experiment. And in natural biological system also, we many a time find equivalent circuit which are equivalent to this type of three gene network. Now, this type of circuit has a particular name. It is called repressilator. Why it is called repressilator? It will be clear as we keep on discussing today about the behavior of this model as we analyze this model using ODE based equations. So, let us start making the model for this system. Here in this model, I will consider mRNA and the protein for each of these three genes separately. So, I will have a mRNA for X, I will have a mRNA for Y, I will have a mRNA for Z, three mRNA, and I will have three proteins X, Y and Z. That means I will have 6 different ODEs. So, for the first one, for the mRNA of X, let us consider mRNA of X is mx. So, that is mRNA of X. I will use the same convention. My will be the mRNA of Y and mz will be the mRNA of Z. So, if I write the first ODE for rate of change in concentration of mRNA of X, that is dmx dt, that will be equal to what I have written here. Look at the equation carefully. K0 plus K1 into a Hill function minus K2 into mx. So, let us look into each of these things carefully. What are they? The first one is for leaky expression. If you remember, many promoter, like even a lag promoter, is not a very tight promoter. It is a leaky promoter. That means even in absence of the control molecule, there will be always a sustained expression of the gene, maybe at a very low level. So, I have considered a leaky expression here for gene X, that is K0. The next term is K1, that is the maximum rate of transcription. And in this bracket, I have shown a Hill function, which is nothing but a negative Hill function. Why I have considered negative Hill function? Because usually this type of inhibitory system where a transcription factor comes and binds to a promoter, this inhibition or even in case of activation, the behaviors are nonlinear as we have discussed earlier, and many a time this nonlinear behavior follows a sigmoidal pattern where initially when the inhibitor is less, expression will be very high, and when the inhibitor increases, there is a drop of rate of transcription in a sigmoidal fashion. And if you remember, we have discussed that an inverse Hill function is a very good simple function to capture this behavior. So, I have considered a Hill function using Z here. Z is the inhibitor because Z is inhibiting X. So as you see in the diagram, Z is inhibiting X here. So Z divided by H is my Hill constant to the power n. So 1 by 1 plus Z divided by H to the power n is my negative Hill function. This K2 into mx is nothing but rate of degradation, that is first order degradation. So K2 is the rate constant for degradation and it is multiplied with the concentration of mRNA. So that's all for my ODE for mRNA of X. Let me clear it up so that we can write the ODE for the protein. For ODE of the protein, it will be much easier. So dx dt, that is the rate of change in concentration of X, depends upon how much mRNA of X is there. So K3 into mx, K3 is the rate of translation, rate constant for translation into a concentration of the mRNA of X minus K4 into X, that is the first order decay or degradation of the protein X. So these two are my ODE for mRNA of X and protein of X. So this is for mRNA and this is for my protein. We will have similar set of two ODEs for Y and Z, and let us consider that all the parameters K0, K1, Hill constant, Hill coefficient, rate constant for degradation are all equal for these three genes. That will make our life easier to analyze. You can consider different value further and see the behavior. For the time being, let us consider that the parameters K0, K1 and other are identical for all the genes. So essentially, I will have triplicate of this set of equation. So for the mRNA of Y, I will have dmy dt, that is exactly similar to dmx dt. Only difference is you have the X here, because X is inhibiting Y. So in the Hill function you have X, and obviously in the degradation term you have my. Similarly for the mRNA of Z, I have Y in the Hill function because Y is inhibiting Z, and mz here is for the degradation part. For the protein Y, dy dt is similar, the equation is similar to dx dt. Only thing is that after K3 it is multiplied with mRNA concentration of Y and the degradation rate is K4 into Y. Similarly, dz dt is for rate of change of concentration of Z. dz dt is similar to dx dt, which is here it is equal to K3 into mz, the concentration of mRNA for Z, minus K4 into Z, the concentration of the protein Z. So that are my 6 ODE, almost similar to each other, and all the parameters I have kept identical for all the 3 genes to make our life a bit easier in this analysis. So if I just brief, K0 is the basal rate of expression which is coming from because of the leaky expression from this promoter. K1 in all cases is the maximum induced rate of expression, that is the maximum rate of transcription when it is induced. K3 is the rate constant for translation. K2 and K4 are rate constant for degradation of mRNA and protein. H is the Hill constant in the inverse Hill function or the negative Hill function, and n is the Hill coefficient. So I have this set of 6 ODEs. As I have 6 ODE, large number of them, I will not try to do any graphical analysis or something. Rather, I will directly go into JSIM and write down the model in JSIM for this particular problem and try to do the simulation with different numerical values and try to understand the behavior of the system. Let us do that. The code for this system is a bit longer and I could not accommodate in one slide. That's why I have broken them in 2 parts. This is the part 1. So it starts with obviously defining time here, and note one important thing, Tmax I have considered as 1000, that's a bit higher, and in this particular model sometime we may have to take Tmax as the maximum duration of simulation even to 10,000 to understand the behavior of the system. I have 6 variables, 3 for mRNA, that is mx, my and mz, these are dependent variables, and 3 for protein x, y and z, and I have a set of parameter values here, and we will play around with these parameters so that we can understand the dynamics of this system at different parameter regime. Once you have defined, this is the second part, the continuation of the code. So once you have defined the parameter, we have to define the initial condition. And beforehand, let me tell you, we will show that this system is very sensitive to initial condition, where you are starting at. So we may have to vary at x, what is the value of x, value of y, value of z at t equal to 0. Here what I have done, I have considered all mRNA at 0, x the protein at 0, z the protein I have kept at 0, y I have kept at 20. And these are my 6 OD based on which the model will be simulated. That's all for the JSIN code. You can try to type that in JSIN and try to simulate as I discuss this one. Let us start look into the results. I have considered initial condition mx, my, mz, all mRNA at t equal to 0 at 0, but all protein x, y, z are equal amount, non-zero values are there. I have taken 20, you can take some other value and check the behavior. So if I have this situation, and then if I allow the system to proceed with time, then what I see, I have 3 plots here for x, y and z. So x will increase slightly, and then it reaches the maximum with a slight peak, and then it falls and reaches a steady state. The same behavior for y. It increases initially slightly, then falls back to a steady state. And then the same thing for z also. It increases initially slightly, then falls to a steady state. What is happening here? As the time progresses, there is leaky expression, there is also initial amount of x, y and z. So initially there is some amount of expression, and that accumulates more and more x, y and z, and then that negative Hill function take control, and it try to pull down the rate of expression, rate of transcription down, and rate of transcription goes down. And after some time, rate of production of mRNA, rate of translation, rate of degradation of mRNA and protein matches, so I get a steady state. So initially there was rise, but as after some time, all of them x, y and z try to replace each other, they all pull them down and reaches a steady state. Now let us simulate the same system with a bit different initial condition. Pay attention to this. At t equal to 0, all mRNA I have considered m equal to 0, but I have imbalance in x, y and z at protein level. So what I have considered? X and z I have kept at 0, they don't exist in the cell. Y I have kept at 20. In the previous simulation, I have x, y and z equal to 20. In this case, only y is equal to 20, x and z are 0. Now if you simulate the thing, you can see a interesting behavior. Here x, y and z, they do not reach a steady state, rather they show oscillation. And the oscillation also have some interesting feature. Initially, the amplitude of oscillation, if you look at near 0 to 200, the amplitude of oscillation is smaller. Then the amplitude increases, it increases further, and then it stabilizes. So x is oscillating with time. Initially it's slow oscillation, less oscillation, amplitude is low, then the amplitude increases, then it further increases, and then it stabilizes at that higher amplitude. The similar behavior is observed for y. Initially lower amplitude, amplitude increase with time, it increases further, and then it reaches a steady value of amplitude. Same thing is for z. So what has happened here? In the earlier case, x, y, z were present in equal amount. So all the repression were equally balanced. So there are equal repression of y by x, that has equivalent repression on z by y, and z has equivalent repression on x. So all are matched together and we got a steady state. But in this case, you have imbalance from the very beginning. So rather than going into a steady state, we get a unstable oscillatory behavior. And that oscillation has an interesting thing. Initially the amplitude is lower, with time the amplitude grows, and then it stabilizes. Let us look further into this amplitude behavior of this oscillation. To show that behavior, what I have done, I have made the phase plot. So if you remember, in phase plot, I will have in one axis one dependent variable, in another axis the other dependent variable. So the same thing, same data, where at t equal to 0, mRNAs were equal to 0, xz was 0, but y at 20, higher value. So here x is in the horizontal axis, y is in the vertical axis. You have started somewhere here, y equal to 20, x equal to 0. With time, we have moved along these. Then with time, our trajectory moved like this, then like this, then like this one. The amplitude is increasing, and slowly we reach this trajectory where the amplitude is much higher. Then eventually we reach this final trajectory where the system keeps on moving in the same loop. The same thing for the x versus z. This x in the horizontal, z in the vertical. You start at that 0, 0 position. You follow this trajectory. Then you come along this. This trajectory takes you to the larger trajectory, so the amplitude of oscillation increases. So as you follow this trajectory mixes with the another new trajectory. You follow this bigger trajectory now, and it increases the amplitude, and eventually you reaches this outer trajectory where it stabilizes. And this outer trajectory in both the cases for xz and xy has the highest, is the biggest one, so the amplitude is the bigger one. And another thing which you may carefully notice here is that the outer trajectory is a closed loop. So what is happening? I have an outer trajectory which is a closed loop. I have started from somewhere here. I entered into one trajectory. That trajectory is not closed loop, rather that was an open loop, and that has taken to me ultimately to this outer trajectory which is a closed loop. This type of behavior is called stable limit cycle oscillation. What happens in case of stable limit cycle oscillation? You have an oscillating system, obviously, as it is oscillate, it is called limit cycle oscillation. The phase plane of that has multiple trajectories. One of the trajectory is actually an isolated closed trajectory. So once you reach that trajectory, you will move around this trajectory, you will not be able to come out. All other trajectory around it are such that they collapse at that closed trajectory. So here, this one, the outer one I have shown as a closed trajectory. And I have started from here, I move along these. I have trajectories here which gives oscillation, but they are not closed. They eventually collapse at this closed trajectory. So as the trajectories slightly away from these closed trajectories are collapsing in this closed trajectory, it is a stable trajectory. This is not a stable steady state, but it is a stable trajectory which will give oscillation. So this type of system is called stable limit cycle, because it is stable because things are collapsing at this closed trajectory, and this limit cycle because initially you have cycle oscillation, but those are not in the closed trajectory but on the open trajectory. So this open trajectory eventually takes the system to a closed trajectory, which is the limit of the whole thing. So it is a closed limit cycle. So it is a limit cycle oscillation. Now let us look into the effect of different parameters in this system on this limit cycle oscillation. How we will do it? Let us first check the effect of the Hill coefficient. Remember, I have kept Hill coefficient same for all the X and Y and Z genes. So I have taken the same initial condition, all mRNA at 0, X and Z at 0, Y is at 20. So that makes the system to give rise limit cycle oscillation. Now I will keep on changing n from 2, initially I have considered n equal to 2, then reduce it to 1.75, then 1.5 and 1. That means what I am doing, I have this type of sigmoidal behavior for n equal to 2, and I am moving towards this one where n equal to 1. So as I move from sigmoidal to hyperbolic, what happens to the behavior of this system? I have only plotted x versus t. You can plot y versus t, z versus t, and you will find the similar behavior. When the Hill coefficient is 2, these green lines, you have limit cycle oscillation, as we have discussed earlier. When I make n equal to 1, I get this stable steady state. I lose the oscillatory behavior. And you can see in this plot, as we are moving from 2 towards 1, from a sigmoidal nonlinear behavior to a hyperbolic behavior and towards a linear one, we are losing this limit cycle oscillation and we are moving towards a steady state. So that's how the promoter nonlinearity controls the existence of limit cycle oscillation in this system. If you remember, in this system model, we have considered leaky expression by K0, right? So in general, when we are talking about limit cycle oscillation, we are keeping K0 at a very low value, 0.03. Now I want to change the value of K0 to simulate a situation where leaky expression is very strong. That means the promoter is quite leaky. So I am changing the value of K0 from 0.03 towards K0 equal to 3. The same system with imbalance in the protein at t equal to 0. When K0 is 0.03, I get this limit cycle oscillation. And as I move toward K0 equal to 3, I get a steady state behavior. I lose the oscillation. That means to get the oscillatory behavior, the promoter control should be very tight. I should not have much leaky expression, because leaky expression nullifies the effect of the repression. Remember, the repressor activity is there in the Hill function, which is separate from the promoter the leaky expression. So if the leaky expression increases with respect to the induced expression, then the effect of the repressor get nullified. So I do not get any oscillation. So in this system, I have to reduce leaky expression to get the limit cycle oscillation. Now, this particular system of oscillation has two other key parameters. One is the stability of mRNA, rate of degradation of mRNA, and the rate of degradation of protein. Rate of degradation of mRNA depends upon the rate constant for degradation of mRNA, and the rate of degradation of protein also depends upon the rate constant for degradation of the protein. So let us look into the effect of mRNA stability and protein stability on the oscillatory behavior of this 3-gene system. What I will do, I have taken the same system where I will get oscillation. In this when I get oscillation, our K2 is at 0.35. Now I want to increase K2. So that's what I am doing. I am increasing K2 towards 0.47, then 1.4. If I increase K2, that means mRNA will become less stable, right? Because K2 is the rate constant for degradation of the mRNA. So if I increase K2, more and more mRNA will get degraded. So as I increasing the rate constant for degradation of mRNA, I can see the amplitude of oscillation is decreasing. That means if I have higher stability of mRNA, then I have higher amplitude of oscillation. And that's quite easy to understand, because I have considered mRNA formation and translation separately. So the translation efficiency, the effective rate of translation, how many proteins will be produced per minute, depends upon how many mRNAs are there. So if the mRNA is very unstable, then the ribosomes will not get much time to produce protein. So if you increase K2, the rate constant for degradation of mRNA becomes very unstable. So the effective rate of production of protein decreases. So my amplitude of oscillation of X, Y, and Z also decreases, because I cannot have higher amount of X and Y and Z. Now let us look into what will happen if I make the protein stable or change the stability of protein. In this case, I am varying K4, the rate constant for degradation of protein, from 0.07 to 0.01. That means what I am doing, I am increasing the stability of protein. 0.07 to 0.01. When K4 is 0.01, it will be much more stable. So if you increase the value of K4 and do the same simulation, you will see when K4 is 0.07, I get this limit cycle oscillation. But when K4 is reduced to 0.001, that means 0.01, that means you have decreased the rate constant for degradation of the protein. That means now the protein will be more stable. I lose this oscillation. Why is it happening? Because if you remember, if I increase X, the extent of repression on Y will increase. Then also extent of repression on Z will also increase, and the repression of Z on X will also increase. This repression, if they are low, then also I don't get oscillation. If the repression is very high, then also I lose the oscillation. So I have to make a balance. And that balance is controlled obviously by how much protein is there in the system. If you make the system such that the proteins are very stable, then this oscillation will get reduced. So higher stability of protein giving rise to lesser oscillation. Now what if I mix both of them together? I want to change the mRNA stability and the protein stability together in the system, and I want to compare with what I have got earlier in a limit cycle oscillation. So I am keeping the initial conditions same where I got limit cycle oscillation. Mx, My, Mz is equal to 0. The mRNAs are 0. X, Z is kept at 0. Y is kept equal to 20. And if I keep all the parameters same as I have shown in the model initially, then I should get a limit cycle oscillation. But what I did in this simulation, I increased K2 to 1.4 from 0.07. Initially K2 was 0.07, and I decreased K4 from 0.35 to 0.04. So I am increasing the K2, that means I am making mRNA less stable. I am decreasing K4, that means I am making the protein more stable. So if I look into the ratio of K4 and K2, I have beta equal to K4 by K2, and that is equal to 0.01. When I had the limit cycle oscillation, initially, my beta was this one. K4 was 0.07 and K2 was 0.35, and the ratio was 0.2. So now beta has changed from 0.2 to 0.01. Essentially, I have made the protein more stable, mRNA less stable. So what type of behavior do I get? If you look into the behavior, if you simulate with this parameter in JSIM and then plot X versus time, Y versus time, Z versus time, I have plotted only X versus time here. You can see initially you have oscillation, but then this oscillation decay and then I reach a steady state, stable steady state. So I have a damped oscillation. Initially I have oscillation, then it gets damped to a steady state. If I draw the phase plane of that, so I have a phase plane plot here with X in the horizontal axis, Y in the vertical axis. So this is my trajectory. Initially things increases, then a oscillation start. The trajectory of these oscillations are collapsing because they are becoming smaller in amplitude. They are slowly collapsing and they eventually collapse at the center of these trajectories, which is a stable steady state. So what I have learned from this particular simulation is that if I increase the stability of the protein, reduce the stability of mRNA, rather than get a limit cycle oscillation, I get a damped oscillation. Let us jot down all the points. So what I have discussed here, we have discussed 3 mutually repressing genes that is forming a repressolator. So it is made up of repressors and it has an oscillating behavior. That's why this type of circuit is called repressolator. Depending on the initial condition and the parameter values, as we have shown discussed earlier, this system can show stable limit cycle oscillation. One key point in this system, if you have to have stable limit cycle oscillation, then there should be some imbalance in the initial concentration of molecules at t equal to 0. Otherwise, you will never get oscillation in this system. We have observed by simulation that if you have higher leaky expression, then you do not get the oscillation. And also we have observed the oscillation require nonlinear sigmoidal behavior in the promoter activity. Otherwise, we lose the oscillation. And finally, one key issue we have discussed through simulation is that the ratio of protein to mRNA stability is a key parameter. If you decrease the ratio, that means you are increasing the stability of the protein, decreasing the stability of the mRNA, rather getting from stable limit cycle, you get a damped oscillation. So based on this relative stability of protein with respect to mRNA, either you will get a damped oscillation or you will get a stable limit cycle oscillation. That's all for this module. Thank you for watching.